Hello everyone, this is going to be two videos I'm going to make for the upcoming diagnostic media lab exam. In fact, I should put that there as diagnostic media lab. But anyway, so this is under tab number 12, which if you're watching this video, you're already there. But I felt like I need to say that anyway. So this first video, I'll go halfway through, probably get through the auger plates, a couple of tests, but I'm just going to basically go through what's written here for you. But on this particular exam, for all the different types of media, basically you have to know what are all the different tests we used, diagnostic media, what is that media selective for, what's it differential for, and then how to interpret the test. And I put here in bold all reagents, and I also put it down here, well, know what each media is selective and differential for, and then down here at the bottom says know how to interpret each diagnostic test that was done in lab and the reagents that were involved. And be able to use that flow chart, and I'll do that in the second video, but be able to use that flow chart to determine some different results, figuring out an unknown. So starting here, some of these tests were selective. Selective means that it only supports the growth of certain organisms. Only certain organisms can grow on selective media. That's what that means. It's going to allow some things to grow. It's going to inhibit others. There were some types that were selective, not very many, but we'll go through those. The differential media means that it has what is called a pH indicator in the media. It's going to change colors if the media becomes acidic. In other words, in the presence of acid, and when bacteria, when they break down sugars for energy, a lot of times it's through fermentation, and fermentation produces acid and gas. So differential means that you can have organisms growing on the same media that are different colors, basically, on the same plate. And I'll go through those two. Know what each type of media is testing for fermentation of certain compounds, such as the disaccharide lactose, like for lactose fermenters. Whether certain organisms produce enzymes, and I put enzyme tests on all those. Know what type of organism. I'm not sure what happened there. I'll fix that by the time I get around. But well, and know what maybe some of the tests are testing for, like how you differentiate between staph and things like that. Know how to interpret each diagnostic test and be able to follow a flow chart. Okay, so here is EMB. Remember we did this one, it was maroon color when it started. I picked one here that had all three results, but basically EMB is a selective and differential media. It is selective for gram negatives, enteric bacteria. That's why the little thing's red there. Basically, enteric bacteria are bacteria from the gut, the mammalian digestive system. And yeah, if you have that in water, it means you have fecally contaminated water. So it is selected for gram negatives, meaning only a gram negative bacteria will grow on it. Now you don't have to know there's a whole category of gram negatives. On your media guide we use to inoculate everything, I put a few there. Escherichia coli, Klebsiella, Serratia, all these others are gram negatives. You don't have to know that. It is differential for lactose fermentation and coliforms. So basically, like this example here, here's positive for breakdown lactose, here's a coliform, here's negative. If you have a lactose fermenter on EMB, it will make what are called purplish colonies, something like that. Kind of a odd purple colony. Negative is the original color, okay? Just it looks something like this, or maybe like that, looks like there's clear colonies. If it's positive for lactose and a coliform, now, coliform is specific. It is from the digestive tract of mammals. It will turn this green metallic sheen or shine on there. So how you interpret this is a coliform, but it's positive for lactose, and it's also positive for being a coliform. Just the purple colonies like that That's not a coliform, but it is positive for lactose fermentation. I had mentioned this in our lab class. Why is this important? Because if you're trying to figure out a bacteria type and you don't know it, only certain things can break down lactose. This is one test that you can use. MAC, or McConkey agar, named after a scientist. It is also, I'm going to bold this, it is selective for gram negatives, like EMB. It's a different color. It's differential for lactose fermentation and coliforms as well. Now, I found this picture. This is what it looks like originally. This is positive for lactose, just sort of a lavender pink colony, really intense. Like here, see how the agar is even stained out here? That's a coliform on McConkey. These clear colonies, or maybe possibly the same color, although they have a tendency to turn clear, is a negative. So positive for lactose, lavender colonies, in other words, pink. Negative for lactose, colorless to pale pink, like that. Positive for lactose and coliforms is like that. Kind of a dark pink, fuchsia, if you will. And like I said, coliforms are a category of bacteria from the mammalian digestive system. Wow. My team scored. All right, so MSA agar, mannitol salt agar. It's kind of a red color. Sometimes it almost looks maybe a little orangish, kind of, but this is basically what it would look like a lot of times when you're starting. This is selective for Staphylococcus. And what that means is only staph is going to grow on this. Only Staphylococcus. It is differential for mannitol fermentation. Mannitol is a plant sugar. It's a disaccharide. Positive mannitol is bright yellow agar. Negative mannitol is the original color. And I use this picture. There are other ones that I could use, but you can definitely see there the entire agar, not only where it's growing, but it will turn the pH indicator here bright yellow. That's a positive. If you had something on this side which looked like it did, that would be a negative. And basically, Staph aureus, and I'll put this on here as well, but Staphylococcus aureus is a bacteria that turns this yellow. Staph epidermidis does not turn it yellow. I'll put that on here when you see this. Blood agar. It is differential for hemolysis. Now, the blood agar, this is actually from our class over here, and then this is an example I just got off of Google, but I used to make this stuff. What you do is you take regular media, you make it, and while it's cooling, when it gets cool enough, you pour sheep's blood, and companies sell it. It comes in a little bottle. It's called defibrillated sheep's blood. It says 5 to 10%, kind of varies. You pour it in there, and of course, it's a blood color. It's red. It's a really pretty red color. You can tell three things from this. You're looking for, do you have a bacteria that can break down red blood cells, which would be pathogenic? There's three things you can get from this, three categories. So it's differential for hemolysis, so breakdown of red blood cells. Gamma hemolysis is no change. It looked like that. You might have growth on the plate, but it's not going to have any change to the color or anything like that. Alpha hemolysis, and we didn't have an example of this, but it kind of gets this green look to it. And it even says here, but the agar is still opaque. Opaque means that something that you cannot see through. Light cannot penetrate it. So in other words, the red blood cells are not broken down completely. There's no clearing like in this one. And beta hemolysis is pathogenic, and that's complete destruction of all red cells. So this is actually a plate from our lab. This is growing on here, and you see the clearing around it. And yeah, you're looking at that, and you go, wait, that's some green agar, or green colored. It really is not. You can see it's kind of a, it's greenish looking, but you can definitely tell. Look at the clearing around where the growth is. And that's what that's supposed to be. It grows like that, and you can kind of see just barely there, but you can basically see through the plate. Like there, you can see the light on the other side. That's not opaque. So opaque means light rays will not penetrate it. That's beta hemolysis, complete destruction of all red blood cells. And yes, that makes it a pathogenic microbe. Okay, agar slants. And we did this, citrate. Citrate agar contains the compound citrate, and all organisms have to have carbon to go through their life cycle and cell division. So once again, why are you doing this? Well, only certain things can do that. Only certain bacteria can break down citrate and use it as a source of carbon. The original color of this media is green. A positive is blue. And what is it positive for? Citrate utilization. That's what you're testing for here. Differential for citrate utilization. That's what the indicator means. So the pH indicator changes color if it can break down citrate. A blue is positive, and a lot of times the entire thing turns blue. It's a very pretty blue. But any blue, like this is the blue slant part, this is still green, that's still a positive. Yeah, negative, it's negative for citrate utilization. It can't use it, so that's the indicator. TSI slants, those are called triple sugar iron agar slants. They start out a red color, or almost even like this color here. What you do with these, remember, you streak the slant, but then you stab the loop all the way to the bottom. You can kind of see a line right there where it went all the way to the bottom. You want the whole thing inoculated. And so it's called triple sugar iron agar. It has iron, it's got 3 sugars. It has glucose, sucrose, and lactose inside the actual agar. So it's differential for that. And it says right there, sucrose, glucose, lactose, and iron. Thus the name triple sugar iron agar. Okay, what is it differential for? Fermenting of any of these sugars, if all or 1 or 2. And for hydrogen sulfide production, that's a toxic gas. It's black colored. It turns the agar black. So positive H2S production, blackened agar. Negative H2S production, no blackening of agar. Negative fermentation, red slant, red butt. Positive glucose only, red slant, yellow butt. And then if it's positive for any fermenting of any of the other sugars, it turns the entire thing yellow. And sometimes it even makes it raise up off of the bottom. So what you can interpret here, this is a negative. See, it's red slant, red butt. This is positive for fermentation of all 3 sugars, or like it says here, glucose, lactose, and or sucrose. So it can be positive for all 3 or glucose and one of the other, either lactose or sucrose. This one produced so much gas that it raised the entire agar up. That's a common result. See, gas production's raising the agar. The lid's closed. So that's just, it's gas production. The blackened here is H2S production. That's a positive. Here is a red slant, yellow butt, and that's for glucose only. That's how you interpret these. So in other words, you need to be able to look at this. What does this test result indicate? This organism is positive for glucose fermentation only. Is it positive for H2S? Yes. Is this one positive for fermentation of all 3 sugars or at least 2 of them? Yes. This is a negative. Phenol red carbohydrate fermentation broth. They start out this color. If you look really close, you can see, oops, I got a little too close. This little tube inside is called a Durham tube. Just think of the movie Bull Durham. And that is to capture gas. Microbiologists are obsessed with gas production. So this is a fluid media. That's what it means by broth. It is phenol red. It's a red color when it starts. This media has small Durham tubes inside for gas capture. Can reveal any gas production from the fermentation of the sugars inside. Now, these do not actually have all of these sugars. You have one type of sugar, but you can get all different kinds. Can have many different carbohydrates that can be used, but it's only one. So I might add that. It's not like the TSI, well, there's three. There's usually one in here, but it's lactose, sucrose, maltose. Those are all disaccharides. Mannitol is a disaccharide. It means two sugars joined together. Glucose is a single sugar, but you can actually have others beside these two. So if the organism can break down the sugar inside, it will turn this yellow. So differential for carbohydrate fermentation, sugar, and gas production. Positive fermentation is yellow broth like this here. Negative is red. This would be a negative. A weak positive for this is orange colored. So it's positive, but it's a weak positive. So it's between yellow and red. Positive for gas production, bubble in the Durham tube, like that one right there is. See that little clearing at the top? See, this one doesn't have that. So it's positive for gas. Negative gas production, no bubble. These two would be negative. This one, you can barely see the tube, and I can't tell enough to see if there's a bubble at the top. But this is positive for gas. These two are not. Okay, and I will start there with the next video.